emptying the entire sea full of its population of fish. So people had this genius idea to come up with fish farms, which is a good solution, but also at the same time there are too many fish in a small area, and that's how fish are not meant to live. So they can get infections very easily, and so these infections will spread like wildfire throughout the fish population. So this is another method that we get protozoal infections, but also helminth infections are probably more common than we get from fish. But fish, it will kill out the whole fish population of a fish farm. So then they give them antiparasitics, or they give them antibiotics, and then it's a whole big problem, right? So I just wanted to bring that up just because I thought it was interesting and something to think about. So then we've talked about a lot about parasites, but one thing I wanted to bring up, does everybody have it? This isn't not quite yet. One thing I wanted to bring up is that protozoa, you know, you're welcome to, when we get to the protozoal section, you're welcome to go do a little scoop from a pond nearby your house, and we can look and see if we can find protozoa in there and see what's in there. They're very common. Not the parasitic protozoa are not extremely common in nature, like wild places that we would drink our water from. But that's basically why we don't drink water from algae-ridden pond, is because it would make us sick, because there are protozoa in there, and if there's too much algae or something like that, that can make us sick as well. Is everybody? Oh, maybe I'll just wait here for a few minutes. So they live in all kinds of habitats all over the world, and we would need to come in contact with those protozoa for us to get them. But let's figure out some ways that we would acquire, or your patients would acquire those infections, and how we can prevent that. But also, I'll go into diagnostics in a little bit. But let's talk about how we would acquire that infection, or how your patient would acquire that infection. So this is a good time to go over reservoir, vector, host, and what those terms mean. So when we're talking about the different organisms, what is the reservoir? So in the case of malaria, for example, so I wanted to start off, when we talked about bacteria, keep it very, very simple. When we talked about bacteria, we gave one example of Borrelia burgdorferi was one of the examples that we gave. Borrelia burgdorferi was the bacteria that causes Lyme disease. So it was the deer, remember, it was the deer, then the deer tick, and then the host was the person. So that's how that disease is transmitted, via a biological vector, which was the tick. Another one was the bubonic plague. Was Yersinia pestis is the bacteria that lives inside of a flea. So the reservoir was the flea. Was the reservoir the flea? No, the reservoir is the rat. So the rat carries Yersinia pestis, is bitten by a flea. The flea bites the human and gives the human Yersinia pestis, which was the bubonic plague, the black plague. So that was reservoir, vector, host. Well, things get a little shady when we're talking about when it's not so clear what is the reservoir and what is the host. For example, like malaria. Malaria, Plasmodium falciparum, for example, malaria, the host is the human, the vector is the mosquito, and the reservoir is the human as well. So reservoir is the human, the vector is the mosquito, and the host is the human. So it's not always cut and dry, but I think it's a good idea to kind of go through those. So like, for example, Trichomonas vaginalis is sexually transmitted. The reservoir is the human, the vector is the human, the host is the human. So it's not always clear cut 1, 2, 3. So it's just a good idea to try and think through what is a biological vector, what is a mechanical vector, and how those things can be transmitted person to person. And mostly when you think about what is a reservoir and what is a host. A reservoir is where it generally lives. The host is what it makes sick, but sometimes those can be the same thing. So I know that can be confusing. I'll try not to confuse you when I ask you questions about it. I'll try and keep it clear cut, whether there's 1, 2, 3 where there's a clear reservoir, a clear vector, and a clear host. Yeah.
If the human is both the reservoir and the host, does it have to be an opportunistic protozoa?
Not always,
because it
like
says
trick is not always opportunistic. That's just
but it says
transmitting.
long-term without illness.
Yeah, you can say long-term without illness or commensal, but some people can live with trick for a long time. So or live with like toxoplasmosis for a long time. So it can be the reservoir and they can be the host at the same time. So yeah, it's not really super clear cut. If I ask you questions about it, it'll be clear cut, but I just wanted to bring those terms back up. Reservoir, vector, host, and what those mean when you read about them. Mostly when you read about things like this, when you read about what is a reservoir, you read about what is a host, you'll read about it in like CDC papers. They'll come out with papers that say, there's an outbreak of this disease, this is the host, this is the reservoir, this is where the biological vector, so they'll include that in their literature, so it's good to kind of understand those terms and what they mean. So I'm gonna play this one for you because I think it's so funky.
I know.
You do?
Not personally, but I watched, she's good friends with Nurse John, and I go to some TV shows.
Oh, that's funny. That's awesome. So from what? How do you know her?
Just Instagram.
Instagram? Oh, that's cool.
She's a brain surgeon, right?
I don't know.
Yeah.
Oh, that's awesome.
From using a neti pot, yesterday I presented a case of a 25-year-old female who came to the emergency department complaining of a headache. She has had a sinus infection for a few weeks and has been using her neti pot. Over the past 24 hours, her headaches became increasingly worse to the point where she went to the emergency room. She was worked up, was given medications for a migraine, and was sent home. The next day, her family noticed that she was confused and also running a fever, so they rushed her back into the ER. What's happening? Headaches, fevers, altered mental status, and a history of a recent bacterial infection is concerning for meningitis. Meningitis is where the meninges, or the covering of your brain, can actually become infected. It can cause all of these symptoms and is extremely life-threatening depending on the source. The cause of meningitis can be from many different things, including bacteria, viruses, can even be inflammatory, can be from cancer, and it can also be from other types of bugs like funguses or amoeba. It can eat your brain. There's a type of amoeba that can actually eat your brain, and it's called Naegleria fowleri. Fortunately, it's extremely rare, with only about 3 cases in the U.S. per year. There was just a case that was diagnosed last week in Florida from using a neti pot. How in the world does that happen? The amoeba can live in fresh, warm water, such as lakes, rivers, or streams, and can even be found in some tap water. It causes the infection when that water containing the amoeba goes to the nose, such as using a neti pot or even jumping into water that has this amoeba inside. It can even live in recreational water that doesn't have enough chlorine, such as splash pads, pools, or water parks. When the amoeba enter your nose through the nasal cavity and water, it can actually adhere to the olfactory nerves and cross through the cribriform plate. The cribriform plate is the part of your nasal cavity where your olfactory nerves or your smelling nerves penetrate through your skull base so you can smell. If those amoeba get on your olfactory nerve, it can actually enter the brain through the top of your nose. The death rate from this type of infection is 97%. It can be detected by being found in the cerebrospinal fluid. Treatments have been challenging to identify because it is so rare and so deadly. Anyone with meningitis type symptoms should always be assessed by a healthcare professional. So are neti pots dangerous? The answer is no, but they have to be used properly. You should only use distilled or sterile water, and you can find that in any grocery store. This is the same thought process for CPAP machines. If you only have tap water, you can use tap water, but it should be boiled for at least 1 minute and obviously cooled off before you place it in your nose. Amoeba can't survive in salt water, which is why it's not found in the ocean, and you can always add salt to your nasal rinse. You should not allow water to forcibly go up your nose, such as jumping into a pool or a lake. You don't allow kids to play with hoses or sprinklers without supervision because it can also place water up their nose forcibly. Please know that this is extremely rare, but it is also avoidable. This particular patient did not survive her infection. So stay safe with those neti pots, and I'll see you guys next week, next Sunday.
Okay, so I know that was like, there's not really a reason to talk about Naegleria fowleri or
Yeah,
to discuss it for
can
any
you
reason,
do-
but the reason I brought it up was because I think it's kinda cool. I think it's interesting and crazy how a protozoa can get up and into your brain. Yeah, in Tahoe? You know, I should look that up. Maybe remind me, I wanna look that up, because I do remember that story, but I don't remember, did you guys hear about that?
I think I roughly remember something like that.
Yeah, I remember something about that. I wonder what it was, if that was it. Oh, crazy. Yeah, I'm from Tahoe, scary.
Is it true for those things I've heard, like,
It was it? Naegleria fowleri? It was? Ah, that's scary. I'm sitting in the lake all
Found
the
lakes in
time.
California, guys.
Ah.
Did those-
Yes, plug your nose when you shove in the water.
is it those protozoa, if they can only just do that, shove parts through your nose, right, like swallowing that water?
Yeah, swallowing with it, yeah. Mhm, yeah.
The acidity.
Yeah, and you know, some places, like, if you go swimming in places kind of frequently and then you get the runs, like that, it's from, there's something in the water that you swallowed. So, you know, it's now that you know about it, you know what these organisms are. You know, it could be bacterial, it could be viral, it could be a protozoa that's fecal orally transmitted. So something to think about. And it gets really gross when we get into helminths. Can't wait for that. Okay, so let's talk about protozoa life cycle. So there are some protozoa have multiple stages of their life cycle, but we're only gonna remember two. And we're only gonna remember two because most protozoa only have these two forms. They have a cyst and a trophozoite. So a cyst is how they get from person to person. You know, protozoa are a little bit more fragile than bacteria and viruses. They're a little bit more environmentally unstable. You know, they can't really handle high heat, they can't handle cold, they can't handle, you know, if they're intestinal, they can't be in other parts of the body. It's almost like if you think of a human cell versus a bacteria, how fragile a human cell is versus a bacterial cell. Bacteria are pretty hardy and they're pretty, they're able to transmit more easily, they're able to survive more extreme environments. Where as you get into eukaryotes aren't very good at handling extreme environments. So what protozoa have done evolutionarily over millions of years to be able to be transmitted person to person or animal to person or whatever is through encystment. So now they can form a cyst, so they can form this hard shell. So what other hard shell sort of mechanisms have we talked about before? Let's go through those. What are they? Capsules? Endospores. So capsule is the exopolysaccharide of bacteria around the outside. So yes, they do help with protection, also they help with adherence. Endospores is a huge thing. So bacteria can form endospores, mostly gram-positive. They form decision. So many good things. Those are really good. I just tried.
Somebody already sees zygos.
I'm sorry, no, zygospores. So zygospores is a great way that the Antheridium and Oogonium can share their DNA and form this little seed structure of a fungi that's environmentally resilient. Are there any more that I'm not thinking of? So now we know that there's a cyst. So this is what, in fecal oral transmission, if someone, for example, poops out, I know I'm just gonna be gross for a minute, poops out the trophozoite is pretty environmentally fragile and they don't really last. Like, and never mind like lasting in a pile of feces and like, yes, there's nutrients there, but you know how generally things don't eat feces. Some flies do or some organisms do, but how do they get transmitted? So they go through this process called encystment, so, or encystation. No, it's encystment and excistation is the two ones that I always get confused between. But it can go from cyst to trophozoite, so if somebody swallows the cyst, then it will enter the trophozoite stage, and once it's in its area where it's happy, then it will turn back into a trophozoite. And then once it's in an environment where it's not happy, then it will encyst. It will gain a harder outer shell and form like an incubation period almost, where it can survive extremes and it can survive without liquid, it can survive without nutrients, things like that. Same as a bacterial endospore. Okay, what else? Anything else? So we're gonna, I'll cover this a few times over, and I have some examples of what they look like. So cysts, they're made of a chitin-like material. I don't want you to memorize that actually, because I don't want you to think it has anything to do with chitin. It's not chitin from fungi, but it's just this harder material. It's just a more environmentally stable material. Some people say it's chitin-like, but it's just a more firm outer membrane. They can exist in that quiescent, in that sleeping stage for months. This is for months, which, yes, months is a long time. It's a very long time when you're talking about transmission. But when you're talking about bacteria that form endospores, they can survive since the beginning of time, endospores. So those are different. These ones, maybe not as resilient as bacterial endospores. They can live about 45 minutes just anywhere, like under the fingernails, anywhere randomly that there's no substrate for them to adhere to. They can live for a long time in places that you might come across them. Cysts, sometimes they're called oocysts, but I don't want you to remember that. It's just a cyst, it's fine. So this is some examples of cysts and what those look like. So here's the cyst and trophozoite of Entamoeba histolytica, just throwing that out there. This is just a picture drawing. Here's the cyst and the trophozoite of Giardia lamblia. Here's the cyst and trophozoite of Plasmodium. This is what causes malaria. But malaria, I wanted to throw it out there too. I have a video on it, but malaria goes through crazy stages. All of these life cycles, through in and out of blood cells. It has a lot of different stages, Plasmodium. But we're just gonna remember there's a cyst, there's a trophozoite. Let's go ahead, Dr. Myers.
Is there something that you want us to memorize other than just the trophozoite and cyst, or is there something else we need to be learning from these images?
Nope, that's it. Just pictures of
Okay.
it. Yeah.
Okay, just to confirm my knowledge.
Yeah.
So a cyst is like the endospore of the protozoa.
Yeah.
And then the trophozoite is the live, actually feeding, living.
It's the vegetative form.
Vegetative form. Yes,
Oh,
yes.
it's a vegetative form.
It is the living, feeding, moving, replicating form.
Okay.
Yes.
Normal form.
Normal form.
Okay,
Great. Thank you for clarifying. That's great. Okay, so those cyst, trophozoite, those are the life cycles that we're gonna cover. That's it. We'll just keep it simple. And so that you understand what those are and what they mean and the purpose. What is the purpose of forming a cyst? The purpose of forming a cyst is transmission, is because they need to get from place A to place B. And the environment between place A and place B might be different, so they have to have this environmentally stable form so that they can be transmitted from person to person or animal to person or however they get around. Okay, so I asked this question last time and then I knew I was gonna confuse you and then I threw it out there and then I wanted to retract it back. But do all protozoa have mitochondria? So this is a trick question, so I don't want you to answer it yet, but let's just think about it. So if you Google it, all eukaryotes have mitochondria, but that's not really the case. So there are some exceptions, And I don't want you to know the exceptions, but I just want you to understand why when we talk about aerobic or facultative aerobics or anaerobes, what's going on with the mitochondria when we're talking about eukaryotes. So just hang in there with me. Okay, so aerobic metabolism, we would think of skin, lungs, maybe blood, because there's oxygen on our red blood cells, right? So maybe bloodborne, but we're thinking of skin and lungs for aerobic metabolism. For anaerobic metabolism, you're thinking vagina, intestines, liver, places where they don't have a lot of access to oxygen. So let's talk about which ones are which.
So you're specifically talking about where on the host the infection is?
Yeah,
Okay.
yes. So I like to ask questions about that because that is what you're thinking about when your patient has an infection. When it's bacterial, for example, and your patient has a bacterial infection, if they have a deep wound infection or if they have a surface infection, if they have a lung infection, if they have an ocular infection, if they have an ear infection, a lot of times then it will kind of give you this first clue of what the organism is. And same with protozoa, same with fungi. What kind of what organism are we talking about? How do we get to know the organism a little bit better? By just surfacely diagnosing our patient, just the first clue of what that organism might be. So here some protozoa have these organelles that are mitochondria-like. I don't want you to know what they are. I don't even care that you know this. I'm just throwing this out there. That some protozoa have these mitochondria-like organelles that function in similar ways to a mitochondria. Even though they're eukaryotic, they might not have a mitochondria, even though most eukaryotic cells, and if you Google it, all eukaryotic cells do have mitochondria, but some have these little mitochondria-like things. I just wanted to throw that out there. You can read about it. But before I go on to CPE, hold on, let me show you my chart, because I want to go to the chart and then I'm going to come back. Okay, so are they all aerobic or facultative? No, some are anaerobic. So, for example, Entamoeba histolytica is anaerobic. Ballontidium coli is facultative, even though they live in the intestines, they can do both. Giardia lamblia is anaerobic. Trichomonas vaginalis is anaerobic. Cryptosporidium, Cyclospora, those are anaerobic. So how is it that they don't die when they're out in the environment?
Make a cyst.
Yes, cyst, because they can form a cyst. So that's how they can survive. You're okay? Want another one? Okay. There's got good stuff in there. I need more chocolate. That's what I need. I don't need it. I need it like a whole bag, but okay. So they can be anaerobic, but they do have some sort of organelle that handles their ATP in manufacturing if they are anaerobic because of our organelle system. Does that kind of make sense? You can just broadly say, yes, most eukaryotic organisms have a mitochondria, but some are anaerobic. Yeah?
Do you want us to know which ones are anaerobic versus facultative?
I don't want you, let me think about it, because I do want you to understand the tissue-related access to oxygen. That is what I want you to understand. So let me think about it. I'll make sure I am clear about that before the next quiz.
I'm confused. Why in the world would you want mitochondria if you're anaerobic?
Because the way that cells have evolved from prokaryotic cells into eukaryotic cells, now there's a division of labor, so they have to have some sort of division of ATP synthesis. So they are able to, in the cytoplasm, go through the stages of making a little bit of ATP. but they have this little funky organelle that's mitochondria-like that can make them more ATP because eukaryotes need more ATP than a general anaerobe does.
But doesn't that require oxygen?
It doesn't require oxygen. They use other funky things. They use, what do they use? They
Sulfur and
use-
stuff?
Yeah, sulfur. They use other final electron acceptors.
Okay.
Yeah.
When you say it's kind of like how humans have an appendix, they don't necessarily use it much anymore, but they used to.
And that is actually a good example because the appendix, we have figured out it actually does contain a lot of white blood cells. So we've actually figured out it is helpful with our immune system. So yes, we don't use it, but it is useful. But the same with the mitochondria with them is like, yes, they do use it. They just use it in a different way. They use it a little bit differently than we do. But I don't want you to memorize that. I'm just throwing it out there.
Does it produce like a certain amount of ATP?
I don't know that. I think it's species dependent. Yeah, but I don't know very much about other electron acceptors. I think your book covers it a little bit. Covers other final electron acceptors other than oxygen, but I can't remember.
But that work, but the mitochondrial life right now is crucial to that.
To that organism, if that organism has one. If that organism has a hydrogenosome or has a mitosome, they have to have that.
Okay, that is right.
Yes, yeah. Okay, so let's go into CPE. What does CPE stand for? Cytopathic effects. So here's the cytopathic effects of protozoa. So, they can directly invade, like fungi can. So they can directly invade tissues and cause tissue damage to whatever layers of tissue that they're attaching to and that they're digging into. I don't want to say burrowing yet because that's a whole nother thing when we get into helminths, but they're residing within that tissue and can damage that tissue directly. Intracellular invasion. So this is similar to what type of bacteria? Let's try and get a back row example. What's one of our bacteria that are obligate intracellular? It's okay. Chlamydia. Awesome. Yeah, there you go. So chlamydia is obligate intracellular. So then they're infecting a certain cell type. What is an example of a virus that's intracellular? Go ahead, Dr. Harris. HIV, awesome. Did you have another one? HIV, awesome. But that was also kind of a trick question. Why was it a trick question? Because they are all intracellular. Yes, awesome. That was fantastic. Yes. So because they are all intracellular, but not all protozoa are obligate intracellular. Most of them aren't. There's only a couple that will invade a cell and live within a cell. Or some of them have a part of their life cycle within a cell. So that's another example of a cytopathic effect is obligate intracellular invasion, where they're inside of a cell and then they can disrupt that cell, such as like Plasmodium. Plasmodium is malaria. Malaria, they have part of their life cycle inside of red blood cells, and so those red blood cells can be damaged and that person is unable to get enough oxygen to their brain or their tissues, things like that. One thing there's a lot of research going around with malaria about sickle cell anemia. Have you guys heard of sickle cell anemia? So there's a lot of African Americans or people who live in Africa or all around the world who are from African descent that have sickle cell anemia, where their red blood cells are shaped like this, and they're sickled instead of like this. So it's like smushed. And so they don't carry as much oxygen on each erythrocyte, so they can have complications from having sickle cell anemia. But the one benefit of having sickle cell anemia is that they're less likely to have malaria or contract malaria because the protozoa is not able to get into the red blood cell and like reproduce and go through its life cycles within the red blood cell. So it's just interesting. Okay, another one is toxic enzymes. So we've talked about this a few times. Lipases, proteases, I gave as examples. But protozoa secrete all kinds of toxic enzymes that can harm human cells. Don't obviously memorize any of that. I just wanted to throw that out there. I thought that was kind of a cool visual. And then immune evasion. So this is kind of a new one a little bit. So what they can do is, what protozoa are able to do is to alter the immune system. So instead of, let me think if I can put it in perspective from when we talked about immunity bypass. I don't want to confuse you. I'm trying to think if that's similar to that. So what's going on is that protozoa are damaging white blood cells. And they're damaging white blood cells so that those white blood cells can't find and trigger a response to the protozoa. So they're defending themselves against the immune system. That's why our immune system, why we can live so long with a protozoal infection, because our immune system has been compromised some way, that they're affecting the white blood cells. So it gives, I haven't talked about macrophages yet and what macrophages are, but what they're secreting in this example, that I thought this was a really cool paper that I read, was they're secreting some sort of protein, enzyme, chemical that doesn't allow macrophages to travel to the area and recognize them. So they're basically like putting blinders on the white blood cells so that the white blood cells can't find them. So it's just something that I thought was cool to bring up, was immune evasion, that they have these tricks that they can evade the host immune system so that they can perpetuate and reproduce and last longer within their host. So that was it. Those were our three main ones. Okay, so here's all of our organisms that we've talked about. Hopefully there's nothing new. There's only two, these ones that are trophozoite specific that don't insist, but I don't want you, just know that they all have a cis trophozoite. Most of them do. You don't have to know which ones don't. I'll think about metabolism. I think metabolism more, instead of memorizing anaerobic, facultative, aerobic, like what I kind of want you to get from the whole point is tissue specificity, that generally if they're in the intestines, they don't have access to oxygen, so they have to either be facultative or aerobic. If they are Leishmania, for example, they're aerobic, they need access to oxygen, so something like that. But I'll go over it on Thursday. And that is it. So that's just your little chart. So I wanted to spend a couple minutes talking about fecal oral transmission, because this is a huge problem all around the world. Oh, there was a video that I wanted to show you too. If we get time, I'm going to show you the video. Maybe I can show you at the beginning of next class. Rewrite myself a note, because that is a super cool video. It's from this video called The Twin Experiment. I put it on here, but have you guys, I forget what the name of the video is. Has anybody seen that? No, it's a documentary about where they gave food to twins to see what their two different diets to twins, and took their biometrics to see what was the resulting, you know, what their cholesterol and blood pressure and weight gain and all that kind of stuff from eating two different diets. But they show this really crazy example of fecal oral transmission, and it's just something that we don't generally think about very much in this country, but in some parts of our country we do. That's kind of why I wanted to show it. But also, it's not something that we think about because there are lots and lots and lots of people that think about it for us. So people that are in water control and like, who, you know, figure out for our lakes, for example, like Folsom Lake. And Folsom Lake is where, you know, people will hopefully not defecate, but children just do sometimes. And there's goose poop everywhere. And, you know, if there's a farm and the farm feces run off into the water, and then where does that water go? The water goes to the water treatment plant. And so how even though it might be some methods of fecal oral transmission are a little bit more obvious, but some are really not that obvious. So I wanted to go through that. So here's the 7 F's. So we start with feces. There's all kinds, some people say 5 F's, some people say whatever F's, but I liked this 7 F's, and even though it's England, it says they spell it different. But 7 F's are start with feces. So we start with fecal oral, this is feces obviously, but it can be transmitted through fluids, through water of any kind. Feces can get into water. Fingers, this is, you would think, I just did like a little 180 in my brain, because feces, we think about fecal transmission on fingers, in my mind, for children is what I think about, like that don't wipe well, that don't wash their hands, then they're playing on the playground and it's transmitted to other children. But why my brain did a little 180 is because half healthcare practitioners don't wash their hands. So, you know, and patients will have C. diff, will have any kind of oral, fecal orally transmitted infection, and it can be transmitted through healthcare practitioners just as much as it can through children, if not more so, because they're guaranteed to be sick from room to room. Yeah?
Sorry, it's kind of disgusting, but
Yeah.
what we're talking about. So when you say like feces can be found in like the poop of children or whatever,
Yeah.
And then they don't wash their hands and then they put their hands in their mouth or something, how does it even get in the poop and stuff? Like if they're a healthy individual, they'll be fine, right?
Fantastic question.
Or
This is actually, I'm gonna give this a candy question because nobody's ever asked that in the 6 semesters that I've taught this class. So nobody's ever asked, like, okay, there's E. coli in the colon, then why do people get sick from eating it? Like, why are people getting sick? Well, because in the esophagus and in the stomach and in the small intestine, and the small intestine goes crazy, then you have the large intestine that comes around, and E. coli lives in the colon. And so little bits of E. coli can come through on food and on things and things like that, but they like to set up shop in the colon is where they like to be. And if there's a lot of E. coli coming into the upper intestines or into the stomach, someone will be vomiting or diarrhea and not feel good. And that's probably the most significant frequency of getting any sort of bug or any sort of diarrhea. All is because E. coli passes through at a quantity that your body can't deal with, that your body is not happy with. Once it's good to the colon, your body's fine. That's where it should live. So that's what happens, is that poo always has E. coli in it. And so when somebody gets too much of that poo on any kind of fomite or transmitted person to person, and too much gets in the upper intestines, that's when a patient will get sick. Yeah, fantastic question. So why every single one of us have E. coli in our intestines? Why do we get sick when we eat it? Well, one, there are different strains as well. So if some person is carrying a strain of E. coli that they've been carrying their whole life and they're fine with it, and they transmit E. coli to another person who is not okay with that strain, their body will have a reaction to that strain. And then some strains are really dangerous that we try to get rid of, like it's like 015H3 or something like that. I forget the name of the strain of E. coli, but it causes, it produces a shigatoxin. It produces a toxin that actually can cause harm to the gastrointestinal system, and it can cause actually harm to our red blood cells. So it can be extreme depending on the type of E. coli, but also, you know, depending on the organism. So, so that's an example of E. coli. But for example, I don't know 100% if it's in the large intestine where Giardia live. So Giardia attaches to the intestines. I can't remember if it's the large intestine or the small intestine. I think it's the large intestine. But Giardia will live in the intestines, and then if they're pooped out, they'll form cysts. So then another person will be exposed to those cysts, and those cysts will get into the trophozoite form into the intestine of another person as well. So that's a protozoa example. Does that answer your question? Okay, so fingers, flies. So a fly that lands on poo and then goes and lands on somebody's apple, and that person can get an infection from that, depending on, yes, it does depend on our immune system. It always does. Infection always depends on our immune system. Every single time, every infection, always. But some people can get it from one cyst, and some people can get an infection from 20 cysts. And it depends on the organism, and it depends on the person. So that's flies, fields. So there's a lot of feces in fields because of manure that they are spraying on crops, and also from runoff from animals. And then fomites. So fomites is a huge problem around the hospital. If you go and do swabs around a clinic that you work at, it's a great idea to go and check it out and see what's being cleaned and what's not being cleaned. And then foods. So I just read this whole outbreak. There's an outbreak right now from eating McDonald's in where? Did anybody else see that? E. coli. Oh, it was E. coli. It was E. coli at McDonald's, and there's like 100 people. Where was it at? Denver, Idaho, Denver.
It's multiple states right now.
Oh yeah. So that's what happened is there's a strain of E. coli that's making people really, really sick, and it's all over their, it's actually, I think it was like the onions that were produced, that were grown for those burgers for that area. Yeah.
Can you explain how a field is?
Yeah, field is like, it can be like fruits and vegetables, but also fields is just an idea to remember that that's where fruit and vegetables grow, that's where runoff can happen in water. So it's just another thing to think about, because really these all came from the same place, right? They all came from a person or an animal. So where it's almost like a life cycle thing, like following the poo. Where's the poo going? It's going from the animal to the factory farm or to the farm. Then it's on the field, then they take the poo, then they put it all over the fruits and vegetables, then it's on the field, then they pick the fruits and vegetables, they take it to the market. Somebody touches it, somebody touches a pen, somebody puts it in their mouth. You know, it's just like how following, it's where it goes. Okay. So I'll try and bring that next time. Okay, so I'm going to start, you know what I'm gonna do actually is I'm not gonna start on our next section, because I was thinking I was going to keep going on protozoa and talk about life cycle and things like that, talk about the rest of our protozoa lecture. But what I wanna do is break up our unknown project into a little bit more bite-sized pieces. So I'm going to pass this around. I'll just put some over here and some over here. So keep this really nice, yeah? I will give you another one if it's getting thrashed, but just try and keep it nice because we'll just save paper that way. So I'm gonna pass out your unknown bacteria project worksheet. So, our unknown bacteria projects can be tricky. And they can be tricky for a few reasons. One reason that they can be tricky is because I talk for 100,000 hours. So I talk for 100,000 hours, and then I say what's important about the unknown project, and nobody hears me, which I get it. I talk too much, that's part of my job, but it is a lot of information, a lot coming at you. But, do you have an extra one then?
No, I printed one out.
Oh, you printed one out. Oh, awesome. Okay. So what we're doing with our unknown project is you're going to get your unknown test tube, and we'll probably get those today. We'll see if we are going to get those today or not. But what we're going to do is you're going to go through step by step to try to identify what's in your test tube. So everybody, if you could try and give me all of your 100% attention. And I think it might be a good idea to just write on the back of here. If you have a pencil, you can write nicely. If you have one piece of paper that you want to write on, that's fine. But I'll say it, and then, oh no, I'm sorry, you can't write on the back of here. You need this. You need a separate piece of paper. But you're going to forget what I'm telling you right now, and then you're going to be sad you forgot. So just try and stick with me.